The immune system has a number of defense mechanisms and responses to protect itself against infection. And these defenses include the strong barrier of epithelial cells that are tightly packed together and which provides insulation from the external environment. The sticky mucus that covers our mucosa and which entraps pathogens and prevents their adherence and facilitates their expulsion out of the body. The acidic environment of some tissues which inhibits and kills many pathogens. We also have a powerful army of cells that constantly patrol our body to detect foreign invaders and eliminate them. In addition to these well-known defenses, we also have immune system-related protective proteins that have antimicrobial activity and help protect against entry of both commensal organisms and pathogens into host tissues. These protective proteins could be divided into 4 major groups, including complement proteins, interferons, antibodies, and the antimicrobial proteins and peptides, and we will refer to them as AMPs. Each one of the first 3 groups of protective proteins will be discussed in a separate lecture, and for today's topic we will focus on the 4th group, which is the antimicrobial proteins and peptides. AMPs are defense molecules that function mainly as chemical barriers in our first line of defense, and they play a major role in the innate immune mechanisms that start acting immediately once a pathogen breaches one of the host's anatomical barriers. These defense molecules provide passive protection against a wide variety of pathogens, and they can exert antibacterial, antiviral, and antifungal activity by inhibiting microbial growth, generally by starving them of essential nutrients, or by directly killing them, mainly by disrupting their cell membranes. And they function like locally produced endogenous antibiotics. And the particularly significant thing is that microbes do not appear to develop resistance against AMPs, unlike the case with antibiotic drug use, even though the microbes are exposed to them for long periods of time. Until now, no bacteria has shown to be resistant to AMPs, even the multi-drug resistant ones. AMPs are either produced constitutively by the body, where they are produced routinely and continuously without activation, or are primarily produced responsively due to the presence of an invading pathogen. And they are mainly produced by our barrier tissues, such as that of the skin and the mucosal epithelia, where they can be secreted onto these surfaces, thus providing immediate antimicrobial action on infection. These AMPs play a major role in the chemical barrier of our body fluids or secretions, such as serum, sweat, mucus, tears, saliva, nasal and respiratory secretions, breast milk, urine, synovial fluid, cervicovaginal fluid, seminal fluid, and cerebrospinal fluid. In addition to their presence in our body's mucosal and glandular secretions, AMPs are also produced by virtually all types of leukocytes and could be secreted locally or could be stored and packaged in some phagocytic cell granules, where they contribute to the degradation of phagocytosed microorganisms. Some AMPs could even be produced by our commensal microbiota. We have thousands of different types of AMPs in our body, but in this lecture we will only focus on 12 major ones, which include alpha and beta defensins, Cathelicidin LL-37, Histatin, Dermcidin, Lysozyme, phospholipase A2, RegIII protein, Secretory leukocyte protease inhibitor, Lactoferrin, Lipocalin-2, the S100 proteins Psoriasin and Calprotectin, and the surfactant proteins SPA and SPD. And these AMPs could be divided into two groups, being either peptides or proteins. And the difference between these two groups is that the peptides are generally less than 100 amino acids long. Now we'll discuss each one of these AMPs, beginning with defensins. Defensins are short, cysteine-rich cationic peptides of around 30 to 40 amino acids that could be largely divided into two subfamilies, alpha defensins and beta defensins. Alpha defensins are produced by Paneth cells of the intestine and are often called cryptidins. They are also produced by neutrophils and macrophages and are part of their granules, where they contribute to the killing of phagocytosed microbes. Beta defensins, on the other hand, are produced by various epithelia, including the differentiated keratinocytes in the epidermis and the type II pneumocytes in the lung. In these two locations, beta defensins are packaged into lamellar bodies, which are lipid-rich secretory organelles that release their contents into the extracellular space and contribute to the formation of the watertight lipid sheet, which is present in the epidermis and the pulmonary surfactant layer in the lung. Beta defensins are also produced by the enterocytes in the gastrointestinal tract and can also be made by the urogenital and reproductive tracts and the kidneys. Beta defensins lack the long protease of alpha defensins and are generally produced specifically in response to the presence of microbial products. And the expression of these antimicrobial peptides is induced or enhanced by signaling through pattern recognition receptors. However, both alpha and beta defensins can also be secreted constitutively by epithelial cells in many tissues, and are stored as well in neutrophil granules. Both classes of defensins have antimicrobial activity against a wide range of pathogens, including bacteria, fungi, protozoan parasites, and envelope viruses. Defensins act within minutes to disrupt the cell membranes of these pathogens, and that's largely due to their amphipathic structure consisting of a positively charged region separated from a hydrophobic region. So they interact with the charged surface of the cell membrane of the microorganism and insert their hydrophobic region in the lipid bilayer, which usually contains negatively charged phospholipids. And that will result in the formation of a pore that makes the membrane of the microorganism leaky and loses its vitality. The next group of cationic peptides are cathelicidins. The only known human cathelicidin is Cathelicidin LL-37, which is an alpha helix type antimicrobial peptide. Cathelicidin LL-37 shares many features with defensins. They are positively charged cationic peptides that have a broad-spectrum antimicrobial activity against both gram-positive and gram-negative bacteria. They are produced and secreted constitutively by different types of epithelial cells and leukocytes, and also are stored and packaged in neutrophil granules and are ready to kill phagocytosed pathogens including bacteria, fungi, viruses, and protozoan parasites. They could also be stored and processed in lamellar bodies. Additionally, both defensins and cathelicidins can be induced by signaling through pattern recognition receptors. The next class of antimicrobial peptides are histatins. Histatins are short, histidine-rich cationic peptides that are constitutively produced in the oral cavity by the parotid, sublingual, and submandibular glands and are found in saliva. Histatins exert a broad range of antibacterial as well as antifungal activity. They are potent antifungal peptides that are active against pathogenic fungi, such as Cryptococcus neoformans and Candida albicans. They bind to surface components on fungal cell membranes and enter the cytoplasm, where they interfere with mitochondrial ATP production and have several other harmful effects on these cells. The next antimicrobial peptide is Dermcidin. Dermcidin is the main skin antimicrobial peptide, which is constitutively secreted by eccrine sweat glands and epithelial cells. It has a broad-spectrum antibacterial and antifungal activities, mainly by producing channels in membranes that disrupt ion gradients. Next, we have the antimicrobial protein Lysozyme. Lysozyme is an enzyme that is secreted by different types of epithelial cells and is present in almost all body secretions, such as saliva, tears, breast milk, mucus, sweat, and cervical secretions. It could also be produced by phagocytic cells and could be found in neutrophil granules, where it contributes to the degradation of phagocytic microorganisms. Lysozyme is a glycosidase that is able to break down a specific chemical bond in the cell walls of both gram-positive and gram-negative bacteria, and it disrupts the same bacterial structure that beta-lactams ultimately target. So its antimicrobial activity is largely due to its ability to digest bacterial cell walls, leading to losses in their structural integrity and creating a defect in their peptidoglycan layer. And it is more effective in acting against gram-positive bacteria because their peptidoglycan cell wall is exposed, in contrast to gram-negative bacteria, which have an outer layer of lipopolysaccharides covering the peptidoglycan layer. Next, we have the secretory phospholipase A2. Another important antimicrobial enzyme other than lysozyme that attack chemical features specific to bacterial cell walls is the secretory phospholipase A2. This highly basic enzyme is secreted in tears and saliva and by phagocytes and Paneth cells. Phospholipase A2 has a direct antibacterial function. It can enter the bacterial cell wall to access, modify, and hydrolyze phospholipids in the cell membrane, which will result in the killing of bacteria. It can also indirectly act to promote bacterial clearance by altering the function of leukocytes in a pro-inflammatory fashion. So when these proteins break down phospholipids, they then produce fatty acids and arachidonic acid, which when metabolized can form inflammatory and thrombogenic molecules that are part of the acute phase response that promotes inflammation. Next, we have the RegIII family of proteins. RegIII proteins are C-type lectins, which are carbohydrate-binding proteins that require calcium for binding. RegIII proteins are secreted by Paneth cells and other epithelial cells of the intestine. They bind carbohydrates on bacterial cell walls, preventing them from contacting the intestinal epithelial cells. Additionally, RegIII proteins are directly bactericidal. They can kill bacteria by binding to peptidoglycans and generating pores in their cell membranes. RegIII proteins, similar to lysozymes, preferentially kill gram-positive bacteria in which the peptidoglycan is exposed on the outer surface. In fact, the lipopolysaccharides of gram-negative bacteria inhibits the pore-forming ability of RegIII protein and further enforcing the selectivity of RegIII proteins for gram-positive bacteria. Next is the secretory leukocyte protease inhibitor. SLPI is a highly cationic antimicrobial protein that can be found on skin as well as in mucosal and glandular secretions, especially in saliva and breast milk. SLPI is an inhibitor of serine proteases that has antiproteolytic and antimicrobial activities. In addition to its vital role in protecting epithelial surfaces from attack by endogenous proteolytic enzymes, this antimicrobial protein can block epithelial infections by different types of bacteria, fungi, and most importantly, viruses. The antiviral activity of SLPI is well noticed in physiological conditions rather than artificial ones, and it's likely due to the interference in events that are mediated by protease, such as entry into the host cell and replication of viral genetic material. Two examples of SLPI antiviral activity are seen in human immunodeficiency virus and human papillomavirus. Higher salivary levels of SLPI in breastfed infants were associated with a decreased risk of HIV transmission through breast milk, and that's an indicator of its anti-HIV properties. Also, high levels of SLPI confer protection against human papillomavirus, mainly by blocking HPV uptake and infection of epithelial cells. Next is the antimicrobial protein Lactoferrin. Lactoferrin is an iron-binding protein that is mainly produced by mucosal epithelial cells and is present in most body fluids, especially breast milk. Lactoferrin is also an important component of the granules of the neutrophil granulocytes and can be secreted locally by these cells. Because lactoferrin is an iron-binding protein, it can bind and sequester metal ions needed by many microbes. And due to this iron sequestering activity, lactoferrin can inhibit the proliferation of bacteria that need iron for their growth. So it has an important role in the prevention of bacterial colonization. In addition, this protein also has a direct bactericidal effect because it can destabilize the bacterial cell membrane. Lactoferrin also enhances the antibacterial action of lysozyme by binding to the lipopolysaccharide layer of gram-negative bacteria and altering its permeability and thus giving lysozyme access to the peptidoglycan layer. Next, we have the antimicrobial protein Lipocalin-2. Lipocalin-2 may also be referred to as neutrophil gelatinase-associated lipocalin and is produced in abundance by neutrophils and mucosal epithelial cells. The antimicrobial activity of Lipocalin-2 is similar to that of lactoferrin and mainly involves sequestering metal ions needed for microbial growth. Lipocalin-2 binds to iron siderophores such as enterobactin, which are essential nutrients for the survival and virulence of many bacteria and fungi, thus limiting the availability of iron and basically starving them of this growth essential ingredient. Next, we have the S100 proteins. We have two members of the S100 family of proteins named Psoriasin and Calprotectin. The S100 family are calcium-binding proteins that show potent antimicrobial activity. Psoriasin, or S100A7, is a protein produced by keratinocytes, and it can be found on the surface of the skin secreted by sweat. Psoriasin is a potent antimicrobial protein against different pathogenic bacteria, particularly against E. coli, and that's why human skin is resistant to colonization by E. coli, despite exposure to it from fecal matter resulting from lack of cleanliness or poor sanitation. Calprotectin, on the other hand, is formed of S100A8 and S100A9 peptides, and is produced in high amounts by neutrophils and by T cells, and in lesser amounts by macrophages and epithelial cells. Calprotectin, just like Lactoferrin, can bind and sequester metal ions needed by bacteria and fungi, which limits their growth. It can sequester zinc and manganese from microbes and also sequester magnesium and iron required by microorganisms, and exerts an antimicrobial effect at the site of its production. In contrast to psoriasin, which kills E. coli, the related protein calprotectin kills Staph aureus but not E. coli, and that demonstrates the specificity of AMPs to certain pathogens. Finally, the surfactant proteins. The two surfactant proteins, SPA and SPD, are members of the colectin family of proteins that are produced by the liver and a variety of epithelia. Surfactant proteins A and D are acute phase proteins that are present in the alveolar fluid of the lungs as well as in the secretions of some other mucosal epithelia. In addition to their lubricating properties in the respiratory tract, these proteins help protect the epithelial surfaces of the lung against infection. Surfactants are microbe-binding proteins that can prevent infection by blocking and modifying bacterial surface components. Additionally, they can be found in the blood where they function as opsonins by coating the surfaces of pathogens. And by doing so, they make the pathogens more susceptible to phagocytosis by our macrophage populations. This function of SPA and SPD contributes to the clearance of opportunistic respiratory pathogens, such as Pneumocystis jirovecii, which is one of the main causes of pneumonia in patients with AIDS.